So good afternoon, colleagues. I'm Professor Dina Naidu, the head of School of Physics. I was requested to chair this meeting at short notice. So welcome to Professor Armita Nourmohammad. Welcome to WITS and to South Africa. I just want to say a few words and then I'll introduce the next speaker. So this particular talk, the Nobel in Africa, is a CS initiative in partnership with Stellenbosch University under the auspices of the National Nobel Foundation and the Royal Swedish Academy of Sciences, with funding from the Knut and Alice Wallenberg Foundation. These lectures are organized as part of the Nobel Symposium in Physics on Predictability in Science in the Age of Artificial Intelligence, the first in the Nobel in Africa initiative. We call this the Nobel Symposium series. It's been happening through the country. And I must thank the following organizers, which is CS, NITheCS, and also the University of Witwatersrand. So I'd like to welcome the University of Witwatersrand's DVC in Research and Innovation, Professor Lynn Morris, to introduce the speaker.
Great, thanks very much, Dina. So, okay, everyone here? Yep. Yeah. Okay, good. Welcome, everybody. So this is really an exciting event, and good to see Francesco there behind the screen. Are you there, Francesco?
Yeah, hello, good afternoon. How are you?
Okay, good, wonderful. So welcome everybody to this very exciting event. So, as Dina mentioned, this is an initiative of Stellenbosch University for Advanced Studies, or what's called STIAS. And this is done, with the permission of the Nobel Foundation under the Nobel Symposium wordmark license out to the Royal Swedish Academy of Sciences and the Norwegian Nobel Committee. And this is funded by the Knut and Alice Wallenberg Foundation, who generously fund STIAS and this outreach program. So, while these Nobel Symposiums have been taking place for a long time, in fact, since 1965, this is the first time it's outside of Scandinavia. So we feel very honored to be hosting this here in South Africa. And we're thrilled that they've focusing on physics as a start. And it's really part of a long-term commitment to really nurture future generations of scholars and intellectual leaders on the continent, and to foster ongoing dialogue between Africa's brightest minds and their global counterparts. And so it provides a unique opportunity to showcase advanced research and scholarship on the African continent, and to promote research excellence and collaborative efforts with the rest of the world. So we're really delighted that our first speaker today is Professor Armita Nourmohammad from the University of Washington and the Fred Hutchinson Cancer Research Center in the USA. And her public lecture is going to be on learning the shape of the immune system and protein universe. And we were just chatting before I came here, just about actually how we talk the same language in some ways, because we talk about B cells and T cells and viruses and structures, but from a completely different perspective, because she's actually a physicist, which is why she's giving this talk. And she did her PhD in 2012 on the evolution of gene regulation. It was on theoretical aspects of that, and that was at the University of Cologne. And she then joined Princeton University as a James S. McDonald postdoc fellow, where she started working on immunological problems, particularly the adaptive immune response in HIV and of course SARS now, from a biophysical and an evolutionary perspective. And then in 2017, she joined the Max Planck Society as a Max Planck research group leader and the University of Washington as an assistant professor of physics. Dr. Nourmohammad is a recipient of an NSF Career Award, an NIH Mira Award, and an APSD Bio Early Career Award. And, we were also just chatting earlier about her collaborators, and we actually know quite a lot, had quite a few in common. I think this is going to be a fascinating and very topical talk. So thank you very much for coming, and we look forward to your talk.
Thank you, Liz. Yeah, thanks a lot for this. What I would say, both here in Stellenbosch and Durban, the royal welcome that we got, and it's been an amazing experience to be here, and I hope this won't be the last time, and that we come back again. As I said, I'm a physicist, sort of dipping my feet in biology. These days, a lot in immunology, and so more recently in some structural biology. So the talk will be rather general because I gather there will be a diverse crowd. But if the physicists in the crowd are interested, I'd be happy to go deeper. If the biologists are interested, to my knowledge, I'd be happy to go deeper. So feel free to ask questions at any point. Yeah, so this is the view that we hope to get usually on campus at the University of Washington. This is what we show during our recruitments. We can sometimes get this view. We get it consistently during summer, but usually this is what we have. So I guess you have the same experience, but it's a bit colder there. So yeah, and so let's get to it. Let's start from the immune system. I think all of us, even the physicists in the room, are very familiar with how immune system works these days after 3 years of struggling with COVID. Yeah, so the good actors in the game are immune cells. You heard about them on the news. Bad ones are pathogens, so viruses and pathogenic bacteria. And if things don't work well, we can get sick, right? And so what I'm interested in is the interaction between immune system and pathogens and how that could lead to different outcomes. So the first line of defense when you encounter a pathogen is what we call innate immune system. So it's very unspecific. Your skin surface is part of your innate immunity. It keeps pathogens from getting to your body, but then there are all these unspecific cells in your body could also interact with pathogens and kill them. So it's a very general thing. It's all over your body and it does its job as well as it could. But then invertebrates, including us humans, we have this very amazing system in our body called adaptive immune system. So when the infection sets in, the adaptive immune system kicks in and sort of starts getting activated. So the main actors are B cells and T cells. And we have a very diverse repertoire of these B cell and T cells to counter the diversity of pathogens that we experience in our lifetime. And so as a result of this diversity, we can mount specific responses to these pathogens. So to get a flavor of what the diversity of pathogens are, this is a map. It's a bit dated, but I think it's roughly still the same numbers. So pathogen richness throughout the globe. And so if you sort of focus in South Africa, you get around, 200 different pathogens that we have, sort of significant large quantity, sort of large concentrated frequency pathogens that we have circulating. But, new things pop up here and there. We experience many new things throughout a lifetime. And so the adaptive immune system is the agent that can deal with that in an adaptive way, as you'll see. So how do these adaptive immune cells, how are they diverse? So this is the physics department, should be all right. So yeah, so if you think these are cells, they have receptors on their surfaces. And so effectively each cell has its own receptor. And to first order, you can assume each cell is different from the next cell. And I'll go into details of that and put numbers on this. But yeah, so a cell with a specific receptor can then recognize a pathogen. And so once it triggers a sort of a massive specific recognition, it can trigger an immune response. And there's a whole downstream effect of different cells and signaling molecules that are involved in mounting this response, but it starts with a specific recognition. So to get a little bit deeper, sort of in detail into what B and T cells are, for those of you who are curious, so let's start with B cells. So B cells have these Y-shaped receptors, or they can express these Y-shaped receptors on their surface. they can interact with surface of pathogens, so pathogenic bacteria, viruses, things that float in our blood, for example. So when the receptors get secreted in our blood, they're called antibodies. So I think all of you probably have heard the name antibodies. So these are just secreted B-cell receptors. And yeah, so they recognize what we call epitopes. So exposed surface of these pathogenic agents, right? So B cells interact with surface of pathogens. T cells, on the other hand, interact with things that are inside the pathogen. So what happens is that all of our cells, not T cells, so all our cells in our body present fragments of proteins that are degraded in our cells, and that happens all the time. And most of these proteins are actually proteins from our own body. They're human proteins, right? And so they present these short fragments of proteins on their surface. So on a molecule called MHC, let's not go into detail what it stands for. So there's a molecule on a cell surface, it presents a peptide. And so most of the time it presents a self-peptide, so protein from our cells. And so if that's the case, T cells interact with it, they don't recognize it that well because it's self, or they recognize it as self and they don't do anything. But once in a while, if the cell is infected with a pathogen, the protein of the pathogen can also get chopped up. And so you can present short fragments of that pathogenic protein. And so that's a non-self peptide, and T cells can get activated by recognizing that. So T cells interact with inside of pathogens because if you think about volume to surface ratio, most of the peptides come from the inside, and the B cells interact with the surface. So movies are always great. So let's look at a T cell interacting with a cell that is infected with a pathogen. So here, T cell, look at this T cell in the red, and this is an infected cell. And there's a sort of timer up there that you can see sort of how long it takes for a T cell to kill this pathogen infected cell. So here we go, just follow them. This is what happens. It's in about 20 minutes, T cell basically kills that infected cell. So to put numbers, and I like usually to give time scales and energy scales when I teach biophysics. So let's try to figure out what are the sort of time scales of these interactions. So as you can see, you saw here in this movie, so at the molecular scale, time scales, right? So for killing of an infected cell, it happens sometime of the order of 10 minutes, 20 minutes or so. But then killing one infected cell is not enough, and activating one T cell is not enough either. So you want to coordinate a lot of different T cells so they act together. And so you need communication. And it happens through small molecules. We call them cytokines. You probably have heard them in the context of COVID. And so that communication happens on the order of 1 hour or so. And cells, then the different types of immune cells can take different roles. Some of them become memory, some of them become killing cells, and some of them effector cells and various names. Don't need to go into details. So that differentiation in role happens of the order of 5 hours or so. Then cells proliferate, they increase in number, they grow. So when you have a specific cell, they just replicate to go on after the pathogen. So that takes a few days. And that's usually when you start feeling sick, when you're activating an immune response. So we have time scales of the order of 10 minutes to days, going from molecular to a cellular response. And then you can think about sort of response at the population level. So you have a human population with the whole immune response to a specific pathogen, and then pathogen evolving to escape that immune challenge. So think about COVID and our vaccine immunity or infection immunity. And so that co-evolution, is of the order of months to years usually. So be it COVID, influenza, or other viruses. So this is more or less the time scale we're looking at. And so when we look at the immune system, we need to consider all of them. The other thing that is interesting, and especially for physicists, if we want to sort of think about, how intricate these processes are. there are various things that we need to satisfy. The first thing is when you recognize something, you need to mount a very fast response. It doesn't make sense to wait for a year and then do something, right? So you want to do it before the host gets sick or dies, right? So what happens is that these red ones are antigens from non-self, and the blue ones here representing peptides from human, right, the self. So if you recognize a non-self, the red one, the response time, characteristic time is of about seconds. Whereas for self, the characteristic time should be infinite, because if you respond, then you are mounting an autoimmune response. So it needs to be fast. It also needs to be very sensitive, right? So generally we have way more self molecules, especially at the beginning of an infection, than non-self. So you get infected with one viral particle. So if you look at specific cell surface and then the number of peptides presented on that cell surface. So beginning of infection, you get about 10 or so virus or non-self particles and 10 to the 5 or 10 to the 6 self particles. So you need to be super sensitive to very small numbers, but at the same time, you also need to be specific, because if you make a mistake, you're mounting an autoimmune response. So, and the reason for the problem here is the affinity of self and non-self to usually the T cell receptors. The difference is not huge. T cell, especially T cells, the receptors are generally of low affinity to everything. And so the dissociation time, if you wish, of a receptor to a ligand is a bit more than 5 seconds for non-self and a bit less than 3 seconds for self. So the difference is not huge. So yeah, so we need fast response, we need sensitive, and we need specific. So how do we achieve that? Equilibrium solutions are not good for this, right? Because you kind of trade the specificity for sensitivity and you don't actually get a solution out of it. So there's actually a lot of non-equilibrium processes involved during an immune response. So I didn't put all the buzzwords here, but we have kinetic proofreading and specifically adaptive kinetic proofreading. Some of you are familiar with that. We have a lot of feedback processes in a sort of interaction between a ligand and a receptor. So basically you need to spend a lot of energy to make sure all of these things are working. So it's a non-equilibrium process to make it work. And so a lot of physicists have been working on various aspects of this, including us. And so this is something quite fascinating. Okay. So to sort of summarize our, 5-minute, 10-minute introduction to adaptive immune system, we have a diverse repertoire of receptors. We can mount specific, sensitive, and fast responses using non-equilibrium processes. It develops throughout our lifetime. So if you look at infant adaptive, they get sick very easy because they don't have a good adaptive immunity. They inherit some antibodies from their mother, but generally, as people grow up, as they encounter pathogens in their environment, they develop new antibodies and new receptors and new B cells and new T cells. And so that's why it's called adaptive. So we can recognize pathogens that did not exist when we were born, right? Because we are just generating new receptors as we see them. And also we keep a memory of infections. And so memory is a memory type cell that are slightly modified, the biochemically modified form of cells, so they can really mount much faster responses compared to a novel response. So these are the features of that. And I think that's what makes this process quite fascinating. And with all the data that is coming in, it's very amenable to quantitative analysis and physics type modeling that physicists or mathematicians are more familiar with. Okay. So one of the topics that I'm very interested in is looking at how inflammation is in immune systems organized. So this is a picture that is reflecting abstraction that has been in the field for a while. It's called immune shape space. And so let me walk you through it. This is a dual space. On the top, what I'm showing is the immune shape space, and the bottom is what's called antigenic or pathogenic shape space. So if you're in the top layer, right, you have immune receptors, let's call them antibodies for just simplicity. And so if you have two receptors and two antibodies that are very close in this shape space, that means that they can recognize similar pathogens. And if you have pathogens or antigens that are close to each other in the lower part, they can be recognized by similar antibodies, right? Now, so this, again, this is an abstraction. The meaning of distance is not very well characterized. There are some anecdotal evidence and you can measure it with some data, but we don't have a very good understanding of even how many dimensions the space has, what determines the distance between two receptors. And so that's something that I'm kind of trying to figure out these days, and I'll show some things about it today. But yeah, so I think this is a way of thinking about this problem. And so there's another feature here that you can see, and I mentioned it before, there's a self and non-self. So the idea is that there are actually holes in this immune shape space which are associated with self-reactive receptors, because we don't want self-reactive receptors in our body to cause autoimmunity. Now, this is being debated. There is more and more evidence that we have a lot of self-reactive receptors in our body, all of us, and they're just not being activated because they don't get enough signal. So there's some active regulation to suppress them. But generally, you can think that there can be some holes in there as well. Okay, so the goal is to figure out how to map out this shape, map out this space. And I'm not going to give an answer to you, but this is an inspiration. All right, so I've been sort of looking at various types of data to get at this question. And so the first set of data that I'm going to show you is DNA sequence or RNA sequence data, so immune repertoire sequence data. Yeah. Yeah. So TCR stands for T cell receptor. PCR is B cell receptor. Yeah. So we have, billions of these cells in our body. And these days, what we can do, it's past decade or so, there's been a lot of technological development in this area. So you can collect blood from a person and then run it through some sequencing machine and do some, some things with the blood, run it through a sequencing machine and get a distribution of receptors that are present in that sample. And so this now gives you a sense of the diversity, at least from 1 liter of blood that you're collecting, the diversity that is circulating, a subsample diversity that is circulating in the patient. So you can produce millions of T cell receptors or TCRs or B cell receptors or BCRs from an individual at a given snapshot. You can do it at time course. So in that case, you can look at dynamics of these receptors, let's say in response to an infection. Or if you look at the snapshot, that gives you a history of pathogenic encounters in that individual because you have all this memory circulating there, right? So one thing I've been working on, has been, others as well, has been trying to make sense of these receptor repertoire data and infer something functional there. Yeah. Yeah, that's me being lazy and pasting sequence. Yes, you're very careful. Okay, good. All right, so the question is, how can we quantify this diversity? Even before any infection, what's the level of natural diversity in an individual, uninfected individual or randomly chosen individual on the street who is not dealing with even an infection? Can we characterize, our null model, our expectation of what should we see in a repertoire? Okay, so let's look at a little bit of where these receptors come from. So a receptor is actually a protein, and proteins are made up of DNA in our body, so they are encoded by the DNA in our body, and generally all our cells in the body have the same DNA, somewhat. And so that means that if all these statements were true, you can sense a hint that they're probably not all true. So we need to have, so we have billion receptors. So that means we need to have DNA for a billion receptors in each cell of us, right? So that different immune cells can get different receptors, right? So the idea is every immune cell has one receptor on its own. We have billion of these receptors circulating. All our cells have the same DNA, generally speaking. That's what we learn in high school. And yeah, to encode for all of these receptors, we need billion different genes in our DNA, right? And so just you all know, billion is a large. But yeah, so where would we fit this billion genes in our cells? So in reality, actually, we have 10 to the 4 genes in a DNA in humans. And these are, the DNA is packed up in a nucleus that is the size, that this is 2 micrometer. I don't think for this crowd, I need to explain what micron is. Yeah, your hair is about 100 micron. And so if I were to fit in 10 to the 9 receptor, 10 to the 9 genes in my DNA, that means that my nucleus, instead of being of the order of 1 micron, needs to be of the order of 1 millimeter, right? So a crayon size. And if that's the case, then everything scales up. And that means that our body should be of the size of a giraffe, which is not. So something is wrong with all our arguments here. So basically, this statement is wrong. All our cells do not have the same DNA. Generally, they do, but well, there are also mutations, so let's ignore that, somatic mutations throughout our life, so we can ignore that for a minute. But then T cells are actually exception to this rule. They actually modify their DNA once a cell decides to become a B cell or a T cell, and they modify and rearrange these receptors. So as a result of that, there's a piece of this DNA that is very specific to this B cells as of the rearrangement. And you can produce a diversity by modifying this DNA. So one thing that we were interested in is to sort of look at the life cycle of B cells and also quantify how this diversity basically comes about. So I'll start with giving you an overview of life cycle and then tell you how we quantify things. So first, you generate the receptor. And this is a very interesting biochemical process, and I'll show you a snapshot in a minute. But for somehow, biochemically, you manage to produce 1 receptor for each cell. And that's before your cell is engaging in any interaction with any type of pathogen. And then for T cells in particular, these cells go through this process that's called thymus, and they get tested. And they, you probably have had, if you want to eat the most clean part of an animal, like the thymus, right? So the thymus is really clean, meaning it only has the self molecules in it. So the tests that happen in thymus is you test these immune receptors, T cell receptors in particular, against all the self molecules. And if they bind really strongly, you throw them out, because that means that later on they also would bind to self receptors and they would save proteins and they would cause autoimmunity. If they don't bind to anything, that means that there's something wrong with these receptors chemically. So you also throw them out. So there is some sort of sweet spot in the middle that you keep the receptors in. So that's the process of thymic selection. Again, this is before any pathogens come into play. So in this stage, you're sending your baby that you generated into school, more or less, to learn how the world works. Then there's a process that you start recognizing pathogens. And so you actually use what you're training to do the job and to kill pathogens, right? And that's a somatic selection. So you then select the good cells to mount a response, you expand clones to mount a response. And that's another level of selection. And then you produce expert cells. And these are the memory repertoires that we deal with, that we keep after we get infected, let's say with COVID, and we keep the good cells to react to. All right. know what happened, to react to a pathogen. Okay. Now, let's see how we generate receptors. So generating these diverse receptors is actually a combinatorial process. It's putting Lego pieces together. So we have a lot of genes called P genes, a bunch of genes, B genes, a bunch of J genes. And if you want to produce a receptor, you choose one B gene, one D gene, one J gene. You insert and delete a bunch of DNA in between, and you produce a receptor. And this is a combinatorial process. And with insertion and deletion, you can actually generate a lot of receptors. And to be specific, you can, the diversity you can generate can be up to 10 to the 23, which is much larger than the number of cells we have in our body. So every one of us, our repertoire is a subsample draw from a possible space of receptors that could be generated. So if I compare even repertoires of two identical twins, because of these combinatorics, they have very different repertoires. So you can model this process using statistical modeling. So you use those receptor sequence data and you build some statistical model that says, you know, what's the probability of using a specific B gene, probability of using a specific J gene, probability of deleting something here, deleting something here, inserting, and you can go through the sort of various scenarios to produce a specific receptor. And so this graphical model, you wish it, you can quantify it, and you can then use it to generate now a repertoire that is statistically similar to something that you're seeing. So this is something that my collaborators here did. And so you can quantify what's the probability of generating a specific receptor and then generate new repertoires or samples to deal with. So this generation process basically gives you a baseline of what a repertoire is before anything functional has happened to it. Now, we talked about this training process, like thymic selection. You throw out, you know, very self-reactive one and non-reactive one, and you keep the good ones in the middle. So usually you keep about 10%. And so to quantify this, we have also developed a statistical model that based on sequence characteristics can tell me what's the probability of a receptor being functional. And so, yeah, so you can build linear models, you can build deep neural networks. I'm not going to go into detail of it, but overall, what you're quantifying is probability of seeing a specific sequence in a repertoire with a given function. And you can do that. So this is what these probability distributions usually look like. If I look at a repertoire, you can quantify this generation probability. And you can look at the distribution of generation probability, usually this is log scale. And yeah, so you can see, you know, the receptors that you're sampling, how easy it is to generate them or how difficult it is to generate them. And what you end up getting is that there's a good share of easy generating receptors, so high probability. And by that, I mean 1 in a million probability, so not super high. And then there's a very long tail of rare receptors that is very specific to one person. So if you compare two repertoires with each other, the things that are common among them are these high probability ones. And there's a whole set of private receptors that would then distinguish you from the next person. So, and if you were to sort of put some intuition on this, 1 in a million would be probability of giving birth to a quadruplet, right? So it's not that common, right? Yes, and so you can characterize these distributions and you can really quantify whatever you're seeing in the data based on these models. And so we wanted to do that. We wanted to see what we can do, what we can ask about the function of the repertoires given these probability distributions. So I'm going to give you some sort of examples here, and there's a lot more. So I mentioned the process of differentiation, that cells differentiate into different functions. So you can start from the progenitor of a T cell, then the T cell can differentiate, become a helper T cell or a killer T cell. Helper one can become a conventional helper or regulatory, many different names. There's a lot more. And so you can sort these cells with experiments, and then you can look, if I now give you a receptor, can you tell me, based on these selection models and functional statistical models, then can you tell me which category they come from? And so this is some sort of a functional space, this distance matrix between these subtypes. And so the colors correspond to those colors here. And so this is some pullback libraries or symmetric pullback library distance between the distributions of these cells. And as we see, cells separate based on according to their function, and even more, you can see clusters associated with tissues that they're sampled from. So actually looking at sequences, building models can tell you something about function at this level. Well, like everyone who is in this field, when COVID kicked in, we started looking at SARS-CoV-2 and immune response to SARS-CoV-2. So we wanted to know whether selection measures that we have can tell you anything about repertoires in patients with different disease severities. So here's what the question is. The question is, can we, if you look at the repertoire, can you tell which receptors are specific to COVID? And this is inherently a difficult question because when you do experiments, you know that about 1 or 2 in 1,000 of these receptors are actually functional to responsive to whatever infection you're looking at, even at the peak of infection. So it's not that your whole immune system is doing something. There's a very small fraction of immune cells that are actually reactive to the pathogen. So we are dealing with small numbers. We're dealing with undersampling and a lot of noise. So it's finding a needle in a haystack. And so to do that, of course, we need statistical modeling. So I'm not going to bother you with all the details, but we combine, differential statistics of these receptors based on sequence data, look at the dynamics of receptors, the sharing of these receptor repertoires across patients that have COVID and sharing with already characterized antibodies against COVID. So you put all of these things together, you go from 1 million sequence down to about 40 potentially reactive sequences to SARS-CoV-2, which then our experimental friends started sort of measuring. So this is all trying to find, signal in very noisy data. And so this has been sort of my receptor repertoire adventures to get at these shape space. But as you know, you can measure some distances in the repertoire. You can try to sort based on function and various things, but we haven't been really successful in characterizing the shape space from sequences. Now I'm going to switch gear and tell you a little bit about protein structures and what we are doing these days and how to characterize that. But before that, if anyone has questions on what I just talked about, feel free to ask. All right, so these receptors, as I mentioned, they're proteins. So proteins are usually, when you want to show them, you draw these ribbons and all this. So that's some schematic way of showing this. So you have antibodies that are interacting with the pathogens. We already talked about that. And then T-cell receptors that can interact with antigens, this little line here that is presented on MHC molecule. And so if I want to understand immune shape space, I need to understand the question of protein shape space. So what's the distance between 2 proteins? And so how functionally distinct are 2 proteins that we have to us, right? And so this is a long-standing problem. Understanding protein structures is a complicated problem. What is a protein? And then we're going to get into some analytical results of how to answer this question. So all of you probably thinking about protein as physicists, think about this, right? The protein-rich food. But in reality, proteins are actually nanomachines that walk and do functions in our body. So this is a protein walking on a microtubule in your cell, so nanometers scale. And so they do fantastic things. Yeah, as I said, they walk around, they mend stuff, they open stuff. So they really, they're out of equilibrium machines in our body. And in reality, as a sort of, before I got into protein structure, I only knew proteins as sequence of letters, 20 letters of amino acids. And that's good when you're, doing analysis at the sequence level. But in reality, there are molecules so these amino acids are molecules that contain atoms. So, there's a real atomic structure here. And so this is now a bunch of amino acids before. So generally, amino acids have backbones, and it's the same for all amino acids, but then they have this thing called side chain, so specific atoms here. And so that is what distinguishes one amino acid from the other. And so usually what happens is that you start from amino acid sequence, so one-dimensional array, which then can fold into a three-dimensional structure. And so that's a sort of self-organized activity. It happens in our cell, shorter than a minute. And so once you have a three-dimensional structure, that determines the function of the protein. So what other proteins it interacts with. So it's all in this sort of, what's the shape of this protein? What's the composition of the atoms and how it interacts with the environment? So going from the sequence to a structure has been a 50-year challenge. It's called the protein folding challenge. And you probably have heard the AlphaFold, about how AlphaFold, that a few years ago, AlphaFold from DeepMind actually solved this problem using machine learning. So this is the protein structure that AlphaFold using sort of very sophisticated machine learning algorithm managed to fold. And so now we have a whole human proteome actually being folded by AlphaFold and being accessible. So machine learning has come to the forefront of protein science. So this is the sequence to structure map. A lot of people also try to use ideas from natural language processing to analyze proteins. So natural language processing is the thing we use when we Google anything online, right? It's this machine learning algorithms to characterize the distribution of words in natural languages. And so one picture that I like to show when I talk about this, which is incorrect, but it's good for intuition, is you want to find some sort of low-dimensional embedding of the words. So if you have a two-dimensional vector embedding of the words, so let's say you have a vector associated with man and then royal, if you sum it up, you should get a vector king, and then woman plus royal should give you a queen. So that's the idea of this low-dimensional embedding for languages. Now, a lot of people thought, okay, we can now treat sequences of proteins as languages. Instead of human language, now it would be amino acid language. Can we try to understand, use these methods to predict anything about protein function? And the answer is yes. They have done amazing jobs in predicting protein evolution, viral escape, and various things. But these models tend to be really large and basically black box, and it's hard to interpret whatever they're doing. So what we were interested in is to start off from protein structure and try to learn function. And the reason for that is that protein structure is much closer to function. So if I go from sequence to function, my model would contain something about sequence to structure map. So it's sort of compound model. And nowadays that we have so many protein structures, we can forget about this part, use AlphaFold to do it. And then try to understand, given a structure, can I predict what that protein does? And that seems to be, maybe an easy problem. We think maybe biochemists should know it or crystallographers should know it, but it's actually a very difficult problem. So unless you work on that protein for your whole life, you probably don't understand when you look at the protein structure, what they do. So to be specific, so here's a concrete problem. So let's say I have an antibody and an antigen. I give you a protein structure, and that means I give you the coordinates and identity of all atoms in both sides. Can you tell me the binding affinity between these two molecules? And can you change some of these atoms to increase the binding affinity? So this is a concrete question, maybe easy, but it's actually difficult to do that. So we were interested to answer this question, not with a black box machine learning, but something that's inspired by physics. And the reason this thing moved, and I was kind of quick in saying this, was I want to have a machine learning algorithm that respects the rotational symmetry in the problem. And why do we have rotational symmetry? If I have a binding energy of two proteins, and if I rotate the two proteins, that binding energy should not change, right? That there's a global rotational symmetry in this problem, right? So you should be independent of the orientation. And if you're training a machine learning algorithm, the machine doesn't know about that. So if you train it all in the one direction, then you give a slightly different direction, it gives you a completely nonsense energy. So you need to constrain your algorithms to respect symmetry. And that's something that is relatively new in the field of machine learning. And it brings in ideas from group theory and very interesting sort of mathematical and theoretical approaches, which we've decided to use to look at protein structures. And so why is symmetry important? I mean, in physics, we all know symmetry plays an important role. There's an analogy between symmetry and conservation laws going back to Armita sort of in her article from 1918 that draws an analogy between time translation symmetry and energy, translation symmetry and momentum, and rotational symmetry and angular momentum. More or less around the same time, we have Albert Einstein talking about principle of covariance. So that's 1916. So in that context, we say laws of nature should be independent of your reference frame. So a reference frame being orientation of my protein. So in this context, we have a law that basically says, if I am a human static with respect to charge, I only measure electric charge. If I'm moving, I measure both electric and magnetic fields. And so if you go to a Minkowski representation, for those of you who know, in that context, you have an equivariance between electricity and magnetism. Same goes between acceleration and gravity. So you can sort of go between the two. And so we wanted to use exactly these ideas to have a covariant machine learning algorithm, and we were able to do that. So I'm not going to go into details of it, but if you're interested, you can read about it here. But so with this algorithm, we were able to learn the statistics of protein microenvironments, if we think are Lego pieces of protein structures. Just someone could tell me how much time I have actually. All right, great. So yeah, so this is your protein structure. One thing we did is we wanted to know if I look at one amino acid in this protein, I mask this amino acid. So meaning I take out all the atoms from that masked amino acid, and then I look at the surrounding of this amino acid. So I take, let's say, 10 angstrom radius around this amino acid. Can I predict what should sit at the middle of that neighborhood, right? And why is this an important question? By constraining, by trying to predict that, you're effectively learning an effective physical potential for that amino acid in the center. And so that gives you, some way of knowing how to configure a neighborhood, and so how to play with the physics of it later. And so we wanted to do it in a rotationally equivariant way. So in a way that you don't depend on the orientation. Yeah, so I'm not going to go into detail. Read the article if you're interested in. We were able to do it. So one thing we did was, all right, we managed to predict amino acids in the neighborhood with very relatively high accuracy, 70% accuracy. And also in a very data efficient way, because compared to other methods, we're not searching the whole space of possible models, but only looking at models that respect rotational symmetry in this case. And so the amino acids that we infer also to be interchangeable, so this other 30% that are not accurately predicted, so the off-diagonal ones. So the ones that cluster together, they actually have common physiochemical properties. So if you see here, hydrophobic amino acids cluster together, of medium size, hydrophobic of small size, means they're clustered together. So in a sense, even our mistakes make sense. And so we check this with evolutionary data, and actually it's consistent with evolutionary variation in proteins. So the model was successful in a sense, it does its job well, and it also reflects physical potential when we test it. And we can try to learn function from it. And so the function we try to do is try to predict the stability effect of these mutations. So this is a protein called T4 lysozyme. So it's a model system for protein structures. And I'm sort of showing two amino acids here. There's a wild type structure, unmutated structure. And then I can look at the mutated structure of these proteins. So here, the orange amino acid to the yellow one is a neutral change. And so it means it doesn't impact the stability. And the purple to pink, the destabilizing mutation. So it destabilizes the protein. And we have about 40 of these with their corresponding protein structures. And okay. And if I look at the predictions that our model makes, so these are all the 40 mutations up here. So here is, for example, amino acid I mutated at position 3 to Y. So that's one mutation. So this is our predictions of our model of the effect of the stability effect of these mutations. And what we see is that generally destabilizing ones are on the red side, on the destable side, and then the neutral ones are on the blue and sort of white ones. And so this is a task that we didn't intend to solve when we trained our network, but because it's a physical network, it's learning something that's similar to energy, and so we can actually solve this problem. And so we can do it even quantitatively by high correlation between the stability effect, experimentally measure the stability effect of mutations and our prediction. So about 70% correlation. So as I said, these are Lego pieces of our protein structures. And so we are now trying to put these Lego pieces together to build up a larger protein interfaces, looking at interaction between, for example, T-cell receptors and antigens, or designing new antigens. And so protein design is actually very interested. We are very interested in designing new antigens. And so the idea that we have is that if we now learn about existing proteins in nature, we can learn how to modify them and build better proteins that do better functions for us. So if you think about this analogy, looking at birds flying, the Wright brothers made a plane. It's not exactly like a bird, but it's inspired by birds. So it has wings, basically. That's about the correspondence, I guess. And so for proteins, it's the same story. So if we look at distribution of natural proteins, the hope is to use machine learning, maybe physical models of machine learning to now build de novo proteins, which is currently actually being done in the lab of David Baker, which is our neighbor in Seattle, for drug delivery, vaccine design, bioactive material design, electronic transport, or many other functions that you can think. So these proteins, they look like coils and traps or various things. They don't look like a real protein, but they follow similar principles, similar molecules are involved. And so what we are really interested in is basically vaccine design for cancer immunotherapy or various sort of antigen or neoantigen designs in that context, and to use that knowledge to go back at the immune shape space story. So I think this is a good ending, and sort of looking forward to the future using these types of model, and so giving you a sense of what's actually possible now and where we are moving. And I think this is a very exciting era to be at. So with this, I'd like to thank all the group members who contributed in some ways in various of these projects and our collaborators and all the funding agencies. And I'd be happy to take any questions.
So colleagues, Armita, thank you very much for an excellent presentation. We learned a lot because we discussed some things like this generally in our meeting this afternoon in physics, by the way.
Great.
So, it was nice to see the cross fields from bio to chemistry to physics, principles and machine learning, which is becoming important for big data. So thank you for the exciting and very interesting talk. So colleagues, I got